Good afternoon. Thank you very much, and appreciate you taking your time out to hear about Galena. We are a publicly traded company, so I would refer you to our forward-looking statements. Galena is working in, I think, a bit of a unique environment. Here at this immuno-oncology conference, there's quite a bit of talk about checkpoint inhibitors, of course, and attacking existing cancer. But Galena is working in the area of prevention of recurrence. So once a patient's been treated, their cancer is successfully treated, they have no evidence of disease, we know for a fact that many of those patients are at a high risk for cancer recurrence, and in fact, much of the mortality of cancer does in fact come from their recurrence, much more so than the primary diagnosis in many cases. Galena is developing products, cancer vaccines, for the prevention of these recurrence. We are in phase 3 with our product NeuVax for the prevention of breast cancer recurrence in HER2 1+2+ patients, as well as a number of other trials that I'll walk through here throughout the course of this presentation. Our development pipeline is shown here with our PRESENT trial phase 3 at the top. It is being conducted under a special protocol assessment, and I'm very happy to say that we just received fast track designation late last week. It was announced early this week, so exciting news and recognition on that. Thank you very much. I can appreciate the challenges there. We're also, NeuVax is also being developed in combination in 2 studies in combination with trastuzumab or Herceptin, looking at both a similar patient population to the PRESENT trial in HER2 1+2+ patients, as well as in high-risk HER2 3+ patients. And we're also looking at NeuVax, carrying it earlier into the disease process, looking at ductal carcinoma in situ with the NCI. We expect to start that trial shortly, and I'll go over these trials in a bit more detail a little bit later. We're also looking to develop NeuVax in gastric cancer, HER2 being a validated target in that disease with our partners, Dr. Reddy. We have a second immuno-oncology program, GAL301, which is also a peptide vaccine program targeting folate receptor alpha or folate binding protein, FBP. That is winding up a phase 1/2 trial with some really excellent data that was announced last year at the ESMO conference, and additional data coming up on our 2-year DFS mark at ASCO here over the next couple of days. Before I jump into the science and a little bit of background on the product, let me just comment that the commercial opportunity for this product is significant. We're in phase 3, so we have what we believe is a reasonably well-defined patient population, and the PRESENT patient population is defined by the phase 3 study in the United States, is, we believe, a several billion-dollar market opportunity. And as we look to expand that patient population through additional trials, we think we have a significant market size available to us and a significant unmet medical need within that market, both in the U.S. and outside of the U.S. So it's a very, very large opportunity and very significant upside for us as we move this forward. So to a great degree, we are redefining the standard of care for our target patient population, and in particular looking at the HER2 1+2+ breast cancer patients, following their standard of care, which is typically surgery, chemotherapy, radiation. Assuming it's successful, and most early stage cancers of this type, they are successful, a woman will be declared no evidence of disease. The treatments will be considered successful, and she'll be sent home now essentially to watch and wait. And other than hormonal therapies, there's really no other targeted therapy that's available for these women. If you're HER2 3+ or HER2 positive, you'd be indicated for Herceptin, or maybe Perjeta off-label, but nonetheless, if you're HER2 1+, you're essentially told to go home and watch and wait, and all you can do now is get on with your life and hope that the cancer doesn't recur. And this is the setting in which we're working. So in these patients, node positive, early stage, HER2 1+2+, about 25% of these women will have a recurrence, and when they have a recurrence, the outcome and the prognosis is very poor. And as I said, the majority of fatalities or the mortality really comes from the recurrence. So the sooner we can catch these patients, the better, and if we can prevent the recurrence of these tumors from coming back, the outcome for these patients is much, much higher. Now, while I'm focused on breast cancer, and we'll focus on breast cancer for this presentation, for our work, this is true across the range of oncology. There's 14.5 million cancer survivors according to ASCO, based on their cancer survivorship publication they put out in late 2014. That number is expected to increase. and with the great work and advances that's being done across the range of oncology, but particularly with checkpoint inhibitors and immuno-oncology, I believe we're going to see a significant rise in cancer survivorship across the spectrum. And all these patients will be potentially targets for various therapies to prevent their recurrence, again, where much of the mortality comes from in cancer. So this is a unique area. It's an area that's not many people are working on, so I think it gives Galena a very significant advantage and lead here as we continue to move forward with our programs. So why do we work in the adjuvant setting? Why is a cancer vaccine appropriate for this setting? Well, our belief is that in the minimum residual disease setting, or a setting where a woman, in our case, her immune system is now healthy, she's recovered from her treatments, there's no existing identifiable tumor mass around, that's an ideal setting for the immune system to do its job. Its sensitivity, its specificity can seek out and destroy cold cancer cells. It can take on micrometastases. It can provide long-term protection with boosting and systemic protection. That's a far better situation, a far better setting than asking the immune system to take on an existing tumor mass that has all of the tumor avoidance mechanisms in place. So we believe that the adjuvant setting is the first place to demonstrate the efficacy of these vaccines. Certainly as we learn more, we can begin to expand that setting either in monotherapy or in combination. We are targeting two well-validated targets, HER2 and folate binding protein, again with the trials that I've identified in the pipeline slide earlier. So the T cell is at the center of the immune system attack on cancer. I think the checkpoint inhibitors and success of those products have demonstrated that very, very plainly, that without the T cell it would be hard to make any kind of material progress in attacking cancers. And without T cells, clearly none of these strategies or approaches would make sense, would work out very well. NeuVax is shown to be a very, very potent generator of T cells, of HER2 specific T cells. This has been published numerous times. We've got quite a bit of data from clinical trials. And so our approach is to identify the setting where NeuVax as a monotherapy can show the greatest efficacy and have the greatest clinical impact, and expand from there through combination trials into different settings where we believe this approach should be efficacious. So what is NeuVax? NeuVax is a 9-residue peptide off the extracellular domain from the HER2 protein. It is haplotype restricted. It binds to HLA A2 and A3, as we've demonstrated clinically. We do believe it binds to HLA 24 and 26 and are working on demonstrating that in one of our combination trials. But nonetheless, it is a 9-residue peptide taken directly off the extracellular domain of the HER2 receptor. We have demonstrated that it generates a very potent and significant CD8 T cell response. This has been published a number of times from our phase 2 trial. And you can see that we can generate a T cell population specific for HER2 expressing cells up to about 2% of the total T cell population, which is a rather large immune response. And we've also shown that with boosters, by boosting patients once every 6 months, we can in fact maintain that response over a very long period of time. Our phase 2 trial ran out 5 years, and we were able to maintain that immune response during that period. We've also shown that we can demonstrate both inter and intra-antigenic epitope spreading, thereby spreading the protective immune response beyond that of simply the HER2 antigen. From a safety profile, the product has been shown itself to be very safe to date. Our clinical data from the phase 1/2 trial showed very minor, primarily grade 1 and 2 adverse events, flu-like symptoms, fatigue, and headache. All of these have resolved after 24 to 72 hours. So it's a compound that can be used to treat women in the adjuvant setting, women who are otherwise healthy and want to get on with their lives. And that safety aspect is very important to the setting in which these drugs can be used. The key data from the phase 2 trial is shown here. This is the phase 2 patient population from the phase 2 trial. NeuVax plus GM-CSF versus control patients. Active control patients, and you can see that the control patients had a recurrence rate of about 25%, which is exactly what we expect in line with historical numbers. And yet the drug-treated patient population had statistically significant reduction in DFS at 2 years and 3 years and had no recurrences until late in the trial, getting close to 5 years. So we treated these patients for quite some period of time with excellent results. This patient population defined our special protocol assessment and defined the phase 3 trial, which is shown here. We did complete enrollment in the trial in April of 2015. We targeted 700 patients. We over-enrolled to 758 patients. The patients are treated once a month for 6 months, followed by booster once every 6 months, so a very patient-friendly administration, which again is important. Remember, these women are otherwise healthy, and so finding a drug that has both a safety profile and administration profile, which is conducive to their current status, is very important. We do have an interim safety and futility analysis coming up at the end of this month, and we expect our primary endpoint to be in about 2 years from now in 2018. The schematic for the trial is shown here. It is NeuVax plus GM-CSF versus placebo plus GM-CSF, with the dosing administration shown there on the bottom of the slide. It is a double-blinded, randomized, placebo-controlled trial targeting HER2 1+ and 2+ patients, HLA A2 and A3 positive patients. Again, the primary endpoint is patients have to be on drug for 3 years, and then at 141 events, and the interim analysis is a futility analysis at 70 events. The trial is proceeding very well, and as I said, we publicly announced that we expect to have an interim analysis at the end of June, at the end of this month. The interim analysis is fairly straightforward. It is a futility analysis and a safety analysis based on 70 events. We'll do a clean and a soft lock of the database and compile the data, turn it over to the IDMC, which will conduct the analysis and return back to us essentially a recommendation on a go or no-go based on the futility analysis of the drug. So the trial is going very well, and we are working very hard to demonstrate the efficacy in this secondary prevention setting, as I said. We also believe, based on additional data and additional work, that NeuVax could be very efficacious in other settings, and particularly moving in the primary prevention setting, with the ultimate goal to possibly treat patients in the primary prevention setting. Can we take women who've never had breast cancer, maybe high-risk women, and actually prevent the primary occurrence of that cancer? To that end, we're working with the NIH and MD Anderson in running a trial on DCIS patients, looking to generate an immune response in patients with ductal carcinoma in situ, a very early and traditionally non-invasive cancer. We're also looking at metastatic disease. We're reviewing a number of potential trials in combination with checkpoint inhibitors to see if we can't move later in the disease process to see if the generation of T cells, a very specific and significant number of T cells, would allow a checkpoint inhibitor to be even more potent in various clinical settings. We have a number of collaborations in place and a number of trials ongoing. The PRESENT trial being shown in the top line, but as I said, we have 2 trials in combination with Herceptin. One trial is in HER2 1+, 2+ patients, so a similar patient population to the PRESENT trial. In combination with Herceptin, there is strong data that says mechanistically NeuVax and Herceptin should be very synergistic. Herceptin has been shown to bind to the HER2 molecule and work by antibody-dependent cellular cytotoxicity, internalizing your receptor, which upregulates the HLA molecules on the surface of the cancer cell, thereby presenting more targets for NeuVax-generated CD8 T cells. So there's a mechanistic rationale for this. That trial is enrolling 300 patients and is ongoing. We're also looking at a combination of NeuVax with Herceptin and HER2 3+ patients, so some higher risk patients. That study is being funded by the Department of Defense. And we also, as I said, are looking at the DCIS, and that trial is being funded by the NCI and supported by MD Anderson as well. We hope to enroll the first patient in that trial here shortly, as well as I said earlier, looking at gastric. So a number of studies ongoing with NeuVax to expand its utility and its footprint, both in the primary prevention, secondary prevention area, as well as in potential other settings. So in the few minutes I have left, we have a second product, GAL301. It is a 9-residue peptide that is off the folate receptor alpha, FBP. FBP is highly overexpressed in ovarian endometrial cancers, making it an ideal cancer target, yet not expressed very highly at all in normal tissue. Ovarian cancer has a much higher recurrence rate than breast cancer, so it is an ideal candidate for this setting. We are using it in the same manner, following the standard of care, surgery and chemotherapy, to treat patients to prevent the recurrence of the cancer. And the recent data we showed, I guess recent last year at ESMO, we were running a phase 1/2 dose escalation trial, looking at increasing dose, carrying the optimal dose forward. And what we found is the optimal dose of 1,000 micrograms showed statistically significant improvement in DFS. A little bit of a surprising result considering we were not powered for efficacy, but nonetheless data that was very exciting and very quite stimulating in this area. We have additional data that will be presented at ASCO, our 2-year DFS data in this trial coming out here over the next couple of days. So we have many opportunities in front of us. HER2 is expressed on a wide range of different cancers, as is FBP, and I think this presents us as we move forward and generate the appropriate data to move forward into a wide range of different cancers. Our leadership team is in place. I'm very excited by the addition of Dr. Bijan Najadnik late last year as our Chief Medical Officer. We've a number of milestones in front of us. We reached our 70th event, which triggers our interim analysis earlier this year. We received Fast Track designation, as I said, which we announced this week, so very excited by that. And our biggest milestone this year will be our interim analysis coming up later this month. But we do have additional milestones coming up in both the NeuVax study as well as the GALE 301 and ovarian study. Financially, we have $34 million in the bank at the end of March. We did do a debt financing, which shores up our bank account, so we're in good shape going forward. Our market cap is high 300 million, almost $400 million. And I'd like to end every presentation that I give with this slide. This is the first patient enrolled into the NeuVax PRESENT trial. It's just a reminder in the course of the day-to-day logistical work and financing work and management of clinical trial, but it does remind us as to why we're here to help patients like this, in our case, from the recurrence of their cancer that they fought so hard to defeat the first time, hopefully keep them from ever having to defeat it a second time. So thank you very much, and I'm happy to take any questions.